Dear student, welcome to EPG Pathshala. Today I am going to give lecture on the module Degradation of Insecticides. These are the chemicals which are known to kill insects and pests. Insecticides which are classified as chemicals known to cause imbalance of microbes in the soil, environmental pollution and health hazards depending upon the physical, chemical and biological properties of these chemicals. Further, the microbes which are present in the soil are known to be the scavengers of these chemicals, which depends upon the physical, chemical and biological properties of the soil. Now, we will start with effects of insecticides on soil. As the insecticides penetrates the soil in significant quantities, it directly affects the microbiological aspects of soil that indirectly influence the plant growth. Here some important effects have been listed that are caused by insecticides as alteration in soil microflora, ecological balance, continued applications causing permanent changes in the soil microflora, negative effect on soil fertility and crop yield, reduction in nitrogen fixing soil bacteria such as Rhizobium, Azospirillum, etc., suppression of the activity of nitrifying bacteria such as Nitrosomonas and Nitrobacter by fumigants like ethylene bromide, VAPM, etc. Modification in nitrogen balance of the soil, adverse effect on ammonification in the soil, interference with mycorrhizal symbiosis in plant, modification in the rhizosphere microflora. Now, the persistence of insecticides in the soil. The duration of insecticide persistence in the soil is an important factor influencing the pest management and environmental pollution. If the chemical persistence for longer time period in soil, it may cause certain undesirable changes like accumulation of the chemicals in soil, increasing the toxicity level, assimilation and accumulation by the plants. Next, accumulation in the edible portion of edible plants, products and root crops. Next, erosion and movement with soil particles entering the water streams, causing soil, water and air pollution. The insecticide persistence in soil may vary from weeks to several years as depending on moisture, structure and properties of the components of the insecticides. For example, eldrin may persist for 4 to 15 years. Considering agriculture point of view, a longer insecticide persistence could result in increased absorption and accumulation of these toxic chemicals by plant and produce hazards to living beings. Due to this, a chronic problem of increased accumulation of agricultural chemicals entering the food chain at highly inadmissible level has been reported in countries like India, Pakistan and several other developing countries of the world. Intensive use of chemicals like DDT that is used to control pests, fungicides and disease in crops can persist for longer period and get accumulated in the food chain, causing bioaccumulation, food contamination and health hazards. Therefore, DDT has been banned to be used in agriculture in concern of public health. Biodegradation of insecticide in soil. This process conversion of insecticide into non-toxic compounds aided by microorganism is known as biodegradation. The biodegradation of insecticides depends on biological, physical and chemical processes, but microbes plays a major role in insecticide degradation. The insecticides or chemicals that are resistant to biodegradation and persist in soil for longer time are called recalcitrant. The chemical reactions which involved in biodegradation of insecticides are as first is detoxification. In this process, the insecticide molecule is converted to a non-toxic compound. In this, even a change in side chain of the complex molecules may turn the chemical non-toxic. So, this process is different from degradation. The process of transformation of a complex chemical to a simpler product causing mineralization is called degradation. It is many times considered synonymous with mineralization. For example, therium is transferred to diethylamine by Pseudomonas. Next, conjugation. It is a complex formation or addition reaction in which the complexity of substrate is increased by organisms or the insecticide is combined with cell metabolite. Example, sodium dimethyl dithiocarbamate is degraded by microbial metabolism. The microbes combine the insecticide with an amino acid molecule present in the cell, inactivating the insecticides or the chemicals. Next one is activation. It is the process of conversion of non-toxic substrate into a toxic molecule that affects the pest. For example, phorate insecticide is microbiologically activated in the soil and transformed to give metabolites that are lethal to weeds and insects. Next, spectrum changes of toxicity. Generally, only one type of pest or insect is controlled by insecticide, but sometimes they produce metabolized products that are inhibitory to an entirely dissimilar group of organisms. For example, PCNB, that is pentachloronitrobenzene fungicide, produce chlorinated benzoic acid as metabolites. The factors like soil pH, moisture, temperature, organic matter content, microbial population and solubility of insecticides influence the degradation process. Also, metabolic activities of microbes involved plays a significant role in the insecticide degradation. Majorly all organic insecticides degrade within a shorter period of time, that is within 3 to 6 months under Indian tropical conditions. Now, moving towards techniques for bioremediations. There are different types of techniques. First, intrinsic bioremediation. It is the natural bioremediation process in which the contaminant is degraded by involving indigenous microorganisms, but the degradation rate is very slow. Next, biostimulation. This phenomenon practices addition of nitrogen and phosphorus as stimulants for indigenous microorganisms in the soil. Bioventing. In this process, the gases like oxygen and methane are added or forced into the soil as stimulants to increase microbial activity which degrade the contaminants in the soil. Bioaugmentation. It involves the inoculation or introduction of indigenous microorganisms into the contaminated sites or soil to facilitate the biodegradation process. Next, compositing. In this, the contaminated soils are piled and treated with introduction of aerobic thermophilic microorganisms that degrade the contaminants with periodic physical mixing and moistening to promote the microbial activity. Next is phytoremediation. Plants are used as hyperaccumulators which hyperaccumulate heavy metals through different plant parts or indirectly by microorganisms stimulation in the rhizospheres by plants. Bioremediation. It is the process of detoxification and removal of toxic or unwanted chemicals and contaminants from the soil and other environment by biological weapons in the form of microorganisms. Mineralization. This technique involves the complete conversion of an organic contaminant into its inorganic form by a single species or a group of microorganisms. Now, next we are taking insecticide degradation mechanism. The insecticide present in soil degrade in the environment through physical, biological and chemical phenomenon. The biodegradation of insecticide is influenced by soil properties such as pH, structure, moisture, temperature, organic matter content and the soil microbiota. Microbial degradation and microbial detoxification has been reported as the most promising and cost effective method for the organic pollutant removal, detoxification of insecticides and remediation of soil in the environment. The biotic transformation of insecticides is mediated by the microbiota, while abiotic conversion occurs through chemical and photochemical process and reactions. The structure and the environment conditions by specific degradation reactions for a particular insecticide. The redox gradients in soil, sediments and aquifers occasionally are used to deduce the type of transformation that can occur. In a similar way, photochemical transformation requires sunlight that is available only on the surface layer of lakes or rivers, plants or subsoil layers. Also, accumulation of biodegradation transformation intermediates occurs when the enzyme reaction rates decrease, affecting the production of the intermediates. Insecticide degradation can occur by various mechanisms. Significant role played by physical, chemical and biological agents in insecticides, herbicides and fungicides transformation. Many soil applied insecticides are degraded more rapidly following repeated application at the same time. The transformation processes are oxidation, reduction, conjugation, hydrolysis, isomerization, hydration and cyclization. The molecules of these are degraded to various resultant products that are generally lesser bioactivity as compared to molecule of parent insecticides, but with exceptional cases metabolites showing greater bioactivity have also been observed. Next example is chemical degradation of carbamate insecticide. The carboxyl esterase enzyme adds in the hydrolysis of carboxyl esterase within the active centers of the protein by reversible acylation of the serine residues. Firstly, the substrate gains access to the active site and then serine causes a nucleophilic attack by acylation on the carboxyl carbamate, releasing a stable acylated enzyme and generating a transition state. The factors responsible are complexity of insecticides and non-extractable residues in soil that are called bound residues and are compounds that persist in soil in the form of parent substance or its metabolites after extraction. It and whole of the process it has been shown in the figure showing the pathway of chemical degradation of carbamate. Next is microbial degradation of Carbofuran insecticide. The microorganism metabolize and lead to biodegradation of carbamate insecticide also help to degrade carbofurans. The various isolated bacteria, Pseudomonas, Flavobacterium, Arthobacter, etc. causing the degradation of carbofuran have been characterized in an effort to better understand the bacterial role to remove carbofuran from the environment. Carbofuran is one of the insecticides belonging to the N-methyl carbamate class used extensively in agriculture. It exhibits high mammalian toxicity and has been classified as highly hazardous. Carbofuran was degraded first to carbofuran phenol and the result was degraded to 2-hydroxy-3-phenol by Syphenogomonas species and Arthobacter species. The process the pathway for microbial degradation of Carbofuran insecticide is shown in the figure. Next is the example is the microbial degradation of insecticide of Finamifos. Organophosphate insecticide Finamifos is used for protection of horticulture crops. Pseudomonas putida and Acinetobacter rhizopharia are two isolated bacteria that can rapidly degrade the organophosphate Finamifos in soil. High nematocidal activity is slowly oxidized to its sulfoxides and sulfones. It is also and is equally toxic to non-target vertebrates. Degradation studies of Finamifos in a range of soil showed half-life values ranging from 10 to 85 days. FEN and its oxidation products FSO and FSO2 depict low to moderate inclination for soil adsorption and its accumulation might result in eventual downward movement towards groundwater. When environmental conditions are favorable, FEN could be leached down to groundwater where its persistence could occur. Therefore, certain tools are needed for the decontamination of natural resource by the remnants and oxidation derivatives of chemicals such as FEN. Next is the photodegradation and microbial degradation of endosulfan insecticide. Both alpha and beta endosulfan can fairly undergo photodegradation, but the two major disintegrated products endosulfan sulfate and endosulfan diol are gullible to photolysis. The endosulfan is quite sensitive to alkyl, moisture and acids, produce sulfur dioxide and endosulfan alcohol after slow hydrolysis with intermediate products endosulfan sulfate. The primary product in the soil in endosulfan with lower quantity of endosulfan diol and endosulfan lactone being generated. Fungi are the major microbial community causing degradation of endosulfan. In water, it is highly soluble when compared with other insecticides and has now affinity for lipid, depicting lower biomagnification and accumulation in the food chains. The degradation pathway has been shown in the figure. Next example is of Imidacloprid insecticide. Imidacloprid is a highly efficient insecticide belonging to a novel class of insecticides called chloronicotinoids. It possesses acetylcholine receptors causing almost complete and irreversible blocking of the postsynaptic nicotinergic. It is highly effective on sucking pests such as aphids, leaf and plant hoppers and other pest species. The process of phototransformation in genobiotics can occur by two ways: direct phototransformation through an excited state, indirect phototransformation by photosensitized processes with presence of sensitizers like TiO2 and quenchers. The process has been shown in the figure. Next example is of degradation of organochloride insecticide. The degradation of insecticides depends on biotic and abiotic factors with different degradation rates. Insecticides like DDT and dieldrin are recalcitrant and when applied to soil persist in environment for a long duration with accumulation in food chains for decades. Co-metabolism is the major process causing DDT to biodegrade with requirement of a substitute carbon source with microorganisms growth on substrate and transforming DDT residues without any nutrient or energy derivations. In reducing condition is the major mechanism for the microbial transformation in reductive dechlorination, in which both the OP-DDT and PP-DDT isomers of DDT are converted into DDD. The reaction initiates with the substitution of hydrogen atoms on an aliphatic chlorine. As the degradation proceeds, there are continuous reactions involving reductive dechlorination of DDT. The anaerobic pathway for the transformation of DDT by bacteria has been shown in the figure, according to which the particular insecticide persists in the soil from few weeks to several years. For example, eldrin, it persists from 4 to 15 years. Therefore, their biodegradation processes has to be identified. In this process, the biodegradation process is a method of choice in comparison to other physical chemical processes, because the biodegradation process are considered to be environmentally friendly and they are also cost effective. Therefore, we have also gone through examples in our lecture that biodegradation helps in the how biodegradation helps in the degradation of insecticides. Now, further students, studies on investigation for the mechanism of microorganism and their enzyme degradation is the need of our to preserve our natural resource, that is soil. Thank you.